No worries, it gets confusing with these virtual sessions and in different time zones everywhere. We still have time here, so I'll go ahead and introduce Stephanie Lacour, who is the Bertelli Foundation Chair in Neuroprosthetic Technology in School of Engineering at EPFL, where she leads the Laboratory for Soft Bioelectronic Interfaces. Stephanie, we can see your slides already, so the floor is all yours.
Okay, thank you very much.
Just to let you know, we will still end at 12.
Yes, and I understand. So I'll go fast.
Thank you.
Again, apologies for the issues with the scheduling. So what I would like to talk about is soft technologies that we've developed to design and test and bring forward towards translational research soft BCI, so soft brain-computer interfaces. And the work I will present is really a collaborative work that has been done, that is being conducted at EPFL in our Center for Neuroprosthetics. So first, I would like to give you some of the ingredients that we use to design, to help us design those soft BCIs. And the starting point is really getting into understanding the anatomy and the dynamics of the biological host. And with this, I'd like to just share some of these views of how different the brain is across the mammal kingdoms. And I would like to in particular point to, these are photographs of various brain, and what you can see is that you have at the bottom right-hand side the photo of the rat and the mouse brain, and at the top left corner, we have the photo of the human brain, and you see that the scale is very different. This is very obvious, but also what is very different is the actual topography of the surface of the brain. So if you look at the surface of the rodent brain, it's pretty smooth. There is some curvature, but it's actually overall smooth. Whereas if we observe the human brain, we have a lot of gyrus and sulci, which are actually giving a fairly complex surface that we need to interact with when you're thinking of designing a soft ECoG. So ECoG is an electrocortical electrode array that you aim at positioning immediately as the surface of the cortex. And so here I borrowed some of a table from a recent paper from the group of Thomas Stiglitz, and where here what they've actually put into a table some very useful numbers that you can use when you want to design your ECoG devices. And this is from left to right, various types of animals to conduct, to interface with later on with your device. So from the mouse to the human via non-human primate. And what they've reported is the brain radius of curvature, and then the associated thickness that you will need to actually design a device that will conform such a curvature. And what is important first to note is that, so in the rodents, in the mouse and rat, you have quite tiny brain radius, which is typically 2 to 3 millimeters. Whereas when you go to a similar surface area, or scaling up to the size of the human brain, you reach about 19 millimeter for the radius of curvature. And then my slide went a bit too fast. Here we go. And then the critical element is really when you are the design engineer in order to see, well, how can I actually manipulate materials so that they conform and they comply to this small bending radius. Then you have one parameter that is called the bending stiffness that is actually useful to compute in order to help you design properly your device. And you have two parameters here that are very important, which is E, the elastic modulus, so which will be the mechanical property of the material that carries your implant, and H, the thickness of that material. And the thickness is particularly important because it's to the cube in the equation. So in this bending stiffness, the the lower the bending stiffness, the more conformable your structure is going to be. So you will want to go to thinnest structure and softest materials. And so in practice, you also need to, the equation on the mechanics is one thing, but the second one is how you can manipulate and handle these devices, because the previous equation, we saw that we should do something very thin and very soft, but actually practically it becomes quite challenging to use if you want to implant such device. And what you're seeing here is a photograph of a clinical ECoG, which is sitting at the surface of a mock-up of the human brain. So we made a 3D printed model, if you want, of the human brain. This is now molded, casted into a hydrogel, so it mechanically resembled the structure of the human brain. And if you position if you lay the clinical array at the surface of this brain, you can see that it actually bends a little bit, but it doesn't conform very well the surface. So now if we actually look at the type of material we have at hand when we do this type of microfabrication for our soft ECoG, we typically play with two types of material: elastomers like PDMS or polyimide, which I wrote here as PI, which is a thermoplastic. And by playing with either the thickness of this material, we've actually been exploring how we can have compliance and conformability. And of course, for the polyimide, if you make it very, very thin, you will obtain a structure that will eventually conform very nicely the corrugation or the topography of the brain. But this really starts to happen when you have polyimide membrane that are about 2 micron in thickness. This becomes extremely difficult to handle as such. And if you want to use such thin material, you usually have to have a transient carrier that will dissolve away so that you can manipulate position, and then the structure will conform. The other approach is to actually use softer material, which will allow you to gain a little bit of thickness. And so if we do the same experiment using PDMS here, we can actually have a similar conformability that you would observe for the 2 micron PI, but with a membrane that is now 25 micron in thickness, and this is actually much easier to handle. And so that's an important metric you need to consider when you design these devices. And so I've already mentioned, the elastic properties. So really, what we're aiming is to bring the elastic modulus, the average or effective elastic modulus of the material we can use to build these soft BCIs, but bring them closer to the values of those of the very biological tissue we want to interface to the brain. And then another parameter that is very important is to understand the dynamics, because the static mechanical property are only half of the problem. In many of the application, the device that we are going to use is going to be sitting onto a mobile structure. Of course, at the surface of the cortex, this is not, the displacement of the tissue is not as much as what you would see here on the right-hand side, where you see those two movies shows MRI movement. And here you have on the macroscopic scale, quite demanding stretch and compression of, in this case, the cervical spinal cord when the person is just moving his head. And here on the right-hand side, you see there is much less motion compared to the macro motion here. This is an enhanced phase amplified MRI, but yet you can see that even at the surface of the cortex, you constantly have tiny or micro motion. So that means that you have a device that is sitting at the surface of the brain. It has to actually accommodate and move along with the underlying brain. And so if I look at what we just discussed, is really you need to analyze where you want to position your soft device to understand at which scale you want to interface. So do you want to aim for single neurons activity, so reaching in the micron scale type of devices, or are you happy with a larger interface which will then allow you to collect low field potential, so more compound information from the brain? So this is your application that will pretty much orient your overall design. And then if we want to match the mechanical properties, what we found is that for the cortex, it's good to actually go to material that will match the dura mater, so the skin, the meninges that are sitting at the surface of the cortex. And turns out that it has an elastic modulus that is quite similar to some of the silicone that we have at hand and we use regularly for microfabrication. And then, but the other important metric is that the brain is constantly moving. And so we have to actually also design structures that can accommodate the regular hertz-based pulsing that is simply inherent to the breathing and the cardiac rhythm. The ad hoc stretch here is less relevant for the brain than it is for the rest of the nervous system. So now if I want to, I've presented you, I've given you the sort of the anatomical consideration that we need to account for when we design those soft BCIs. Now let's see how we can engineer then elasticity in electrical and interconnect that we may want to integrate into to form the electrodes. So here is a summary of the approach we're following to build those systems. So we use microfabrication. So we use standard either stencil photolithography or shadow mask or even lift-off photolithography to pattern thin layers of metal. So here what you see is layers of thin gold that we deposit and pattern directly onto the elastomer. The black dots corresponds to our electrode side. Because we use standard patterning techniques, we can pattern arrays of macroscopic electrodes, like the top photo, but we can also miniaturize further the structure, where here we have electrode array that are typically hundreds of micron in diameter. And so depending on where you want to position the array, you can then tailor and adjust the layout of your ECoG. So if you want to cover a large surface area of the cortex, so 5 to 10 centimeters, then we would go for a macro ECoG that is very similar to the layout of the current clinical devices. Or if you want to check or understand more precisely a specific region of the cortex, you may go for smaller devices, typically a centimeter square overall, that will allow you to really explore the activity that is happening in one of the sulcus, for example. So then we spent quite some time optimizing also the reproducibility of the manufacturing of our approach. Here, this is a very extensive number of experiments that we've done to actually get some sort of standard in terms of the reliability and reproducibility of our electrode. The first plots represent our typical impedance at 1 kilohertz for a range of electrode diameter, and we do this over multiple batches of sample. So everything is produced over a wafer, so we make 10 devices per wafer. This is across 160 electrodes. This is the average type of impedance that we have now obtained. So we have a yield of 95% for this type of electrode in terms of reproducibility and stable response. And then the other series of tests, of course, we need to do is what are the mechanical stability of these devices, because we need to evaluate how they're going to perform, or whether the type of deformation they'll have to withstand in vivo, or in particular during implantation, is this going to affect the impedance of the electrode? So we've done various tests where we manipulate the devices in various shapes, like the one shown here on the photograph, and we monitor that the impedance is not affected by that. Then another part that is very important when you're thinking of clinical translation is that the surgeon in particular and the neurologist later on want to be able to see where the device is going to be positioned onto the surface after implantation on the surface of the brain. And what you see here, the top row is a clinical grid, where this is an experiment we had the opportunity to conduct in a human cadaver at the University Hospital here in Geneva. And then we compare the clinical grid that is made of platinum wires and platinum discs, so fairly thick material. And we've done standard CT scans and MRI scan. And here you see clearly the electrode contact showing up from the clinical grid. And then we did the exact same experiment and imaging sequences for a soft grid that has exactly the same electrode layout. So here already you see on the photograph that the array seems to conform much nicely nicer than the standard clinical grid, the surface of the brain. And we also were quite happy to see that our electrodes are clearly visible in the CT scan and do not generate MRI artifact when they're in the scanner. We also tested the foldability of the system. So we selected this particular sulcus where really the implant had to be folded into two. We inserted it into the sulcus, and then we did the imaging, and you see here that now we can see the two layers of electrodes nicely facing each other, which means that we have electrodes that are in contact with both sides of the sulcus. So pushing this soft BCI, we believe, can also, although it's extremely challenging surgically to do this in live situation, it opens ways of interfacing the brain in matter that are not possible today with clinical grid. So we also checked whether in terms of MRI compatibility, we had no extensive heating, and this is summarized in this graph. Just because of I'm looking at the time, I just want to go to the next set of slides, where then we actually use these devices to verify their functionality in acute recording. So what you're looking at here is an acute experiment we did in a minipig, or in a pig actually, where we positioned the ECoG grid at the surface of the cortex, which corresponds to the area of the pig cortex that where we are able to capture information when we stimulate the snout of the animal. So if we stimulate the animal at multiple location, we were able then to record activity at the surface of the cortex. And what you see on the little disc that you see, the big and the little disc that you see here on this photograph, corresponds to a range of electrodes that we've tested, overlaying one on top of the one after the other and of the same surface, in order to compare the type of information we're able to capture going from big electrode to small electrodes. And what we found is that really there's sort of an optimum in terms of dimension, at least in the geometry we've tested, where 300 micron electrode seems to give us the most detailed information compared to the big ones, of course, and there was not so much benefit in going to 250 micron diameter electrode. So that was done acutely, and then the final test we did was to, with a collaboration with the group of Blaise Divert in Grenoble in France, we actually looked at testing one of our electrode array at the surface of the cortex, again, that corresponds to the area of the cortex that can capture some of the activity that is generated when the animal is actually vocalizing. So we learned that mini pigs are actually vocalizing. And what you see here, this was done with a wireless system, so mounted on a head stage directly on the skull of the animal. This is an overlay of the electrode array. And then this is a typical heat map that we recorded. So you have time as a function of the frequency or the potential here that is monitored through the system for one electrode, and this is all of the electrodes overlaid in the system. And you see that we're able to capture with the soft electrode some of this activity that is really specific to the vocalization of the animal. And so with this, I hope I've convinced you that with soft materials, you can actually design and tailor really the layout of the electrode to the zone of interest for the cortical interfaces. But you can also use, of course, the compliance of the system to design it to reach area of the cortex that are not usually easily reached or explored with standard or stiffer technology. Of course, this is a very interdisciplinary research. You need to have inputs from many players, so this is a highly collaborative work. And also what we've learned over the years now is that you really need to align performance of your device with your expectation. And especially when you push towards translational application, you really need to have high reproducibility and stability of your device from one run to the other. And with this, I'd like to thank my lab, and in particular Florian Falliger, who's now a postdoc in the group, but was the PhD student who actually drove the core of the work I presented today on the soft ECoG. I'd like to thank my colleagues who are helping on the implementation, the surgery, and also the data analysis, various sources of funding, and thank you very much for your attention. And again, apology for the delay at the start.
Great, thank you so much, Stephanie. It's really beautiful work, and thank you for finishing on time despite everything.
Thank you.
That's really wonderful. I don't see any